All right, I am Felix Francisco Sanchez, a junior studying mathematics and statistics at Purdue University.
And hello, my name is McKeith Pearson. I am a senior in molecular biology here at Purdue University. And today we're going to be presenting on our presentation, which is titled Quantitative Analysis of Yeast Cell Morphology Defects Induced by Gene Deregulation. Neurodegeneration is the process of neuron cell death. This is a neuron cell. Neurons are arguably one of the most important cells in your body due to their function, which involves thinking, moving, and sensing your environment. Neurons are found all over the body, but most are in the brain. A neurodegenerative disease is a disease that causes massive death of neurons within the brain. There are several neurodegenerative diseases, such as Alzheimer's disease, Parkinson's disease, spinocerebellar ataxia, and finally, the topic of this afternoon's discussion, Huntington's disease. All of these diseases are genetically inherited and progressively fatal with no known cure. So Huntington's disease is a progressively fatal autosomal dominant disorder. Autosomal dominant means that if a couple has a child where one of the parents is affected with Huntington's disease, that child has a 50% chance of getting Huntington's disease. This image depicts a scan of a normal brain on the left, and on the right is a brain of a person affected with Huntington's disease. The dark regions within the brain are supposed to be occupied by neurons. Which isn't showing up. The dark regions are supposed to be occupied by neurons. Symptoms of this disease include loss of motor control, dementia, and mood swings. Huntington's disease is affected by people who are within the ages of 35 to 50 years old. To the left is a description of how a cell makes a protein through a process known as transcription and translation. So transcription involves converting DNA that is known, converting DNA to what is known as a messenger RNA, labeled mRNA. So translation is when that mRNA serves as a template to make the actual protein. The protein is then folded and processed into its proper shape, which is very important for a protein's function. In this example, DNA is encoding what is known as a triplet repeat of nucleotides, cytosine, adenine, and guanine, or CAG for short. This CAG region is then converted into a sequence of amino acids known as polyglutamine regions, or polyQ for short. Research has shown that these polyQ regions play a role in protein-to-protein interactions and protein localization. Many proteins contain polyQ, including ones that play a role in the pathogenesis of Huntington's disease. This is a representation of the Huntingtin protein. The different boxes represent Huntington's modular domains that contribute to its many functions, which include endocytosis, which is how a cell takes in outside material, signal transduction, which is a cell's response to external or an internal chemical signal, and it inhibits apoptosis, which is programmed cell suicide. So, what about Huntington causes Huntington's disease? In a normal protein, an upper limit of 35 glutamine repeats is allowed for normal function. Once that threshold has been exceeded, then you have Huntington's disease. So, how does polyQ overexpansion in Huntingtin protein cause Huntington's disease? So Huntingtin is known to have an interacting partner known as Huntingtin Interacting Protein-1, HIP1 for short. So HIP1 is involved in many functions, including endocytosis and protein trafficking. In a Huntingtin protein with an overexpanded polyQ region, the shape of the Huntingtin protein is believed to be altered, causing the Huntingtin protein to aggregate inside of the cell and become toxic. Some think that this toxicity triggers apoptosis, where cells can opt to commit suicide if they are damaged beyond repair, therefore causing the neurodegeneration. Interestingly, it was observed that HIP1 could no longer bind to a mutant Huntingtin that had undergone polyQ overexpansion. It has also been shown that HIP1 is highly expressed in the brain, making it very likely that both Huntingtin and Huntingtin Interacting Protein-1, or HIP1, play a role in Huntington disease pathogenesis. So just to give you guys a quick summary of what I went over, Huntington's disease is a neurodegenerative disorder. PolyQ overexpansion on the Huntingtin protein causes Huntington's disease. HIP1 is known to bind to Huntingtin, whereas a mutated Huntingtin, which has overexpanded polyQ regions, cannot bind to each other anymore. And then more research needs to be done on the precise mechanism in which polyQ overexpansion inhibits Huntington's interaction with HIP1. So we are now faced with two questions. While working with mammalian cells will be ideal in some situations, it can at other times prove to be more of a hassle. First, mammalian cells are fragile, having a small window of temperature that they can grow in. Second, it is difficult to transform foreign DNA into mammalian cells relative to other model systems. And then lastly, cells have large regions of non-coding DNA known as introns, making their genomes more complex. Yeast have ideal qualities in the areas mammalian cells do not. They are less fragile, transforming foreign DNA into them is relatively easy, and yeast have much simpler genomes. Because of this, yeast is a very common model system for researching molecular pathways that can be found in mammalian cells. In yeast, there exists a protein that does the same function as HIP1 in mammalian cells. This protein is known as yeast HIP1, labeled Y-HIP1. As you can see, they both have very similar regions and both are involved in regulating endocytosis. Now, although there is no known Huntingtin homolog, or there's no functional similarity between Huntingtin and Epsin2 yet, we believe that since Huntingtin contains these polyQ regions, actually more than one, that Epsin2 and Huntingtin are actual functional partners within the yeast cell. Now that we have identified two proteins of interest in the yeast system, we can propose this aim. So our aim is studying the molecular interaction of both Epsin2 and yeast HIP1 in yeast will let us propose that Huntingtin and yeast HIP1 interact in the same way as they do in humans. So, a deregulation of Epsin2 leads to cytokinesis abnormalities. On the left is a normal version of a yeast cell. On the right is an abnormal cell division of the yeast. And similarly in human cells, you have normal cell division, and on the right is abnormal. Okay, so a deregulation of Epsin2 leads to cytokinesis abnormalities, as you can see here. Cytokinesis is the last stage of cell division, where the daughter and the mother cell finally split off. Additionally, cells that have defects when yeast HIP1 is not present in the cells revert back to this normal cell division, as you can see here. Because of this, our lab hypothesizes that Epsin2 interacts with yeast HIP1 to regulate cytokinesis. Just to reiterate the significance of our yeast cell research, our lab has performed the same experiment in human cells. As you can see, in the human cells, cell division was disrupted. These experiments are done with a dye that allows us to see the interior of the cell, called methylene blue. So again, to give you another summary of what I went over, studying Epsin2 and yeast HIP1 interaction in yeast system will allow us to perform more experiments more quickly towards understanding the interaction of Huntingtin and Huntingtin Interacting Protein-1 in mammals. In budding yeast, we're able to overexpress multiple truncations, which are short versions of each protein, that allow us to determine specifically where on Epsin2 and yeast HIP1 cell division regulation happens. Now, to see which regions of Epsin2 and yeast HIP1 are required for regulating cell division, we used a reductionist molecular biology technique known as combinatorial analysis. This involves making shortened versions of proteins called truncations that strategically eliminate regions we suspect are important for cell division. We hypothesize that the combinations of Epsin2 and yeast HIP1 truncations that reverse the cell division defects will contain domains sufficient for regulating cytokinesis. For example, if we were to discover that a particular combination of yeast HIP1 and Epsin2 recovered cell division abnormality, then we can propose that the same interaction is required between Huntingtin and Huntingtin Interacting Protein-1 in human cells. Once we transform yeast with both proteins, we add methylene blue dye, which stains the interior of only dead cells for methylene blue stain microscopy. Then we analyze each image we take. Combining each possible Epsin 2 and yeast tip 1 truncation has given us 5 categories of cell division defects. First obvious is being normal, which is 1 nucleus per cell defect. Then we have what we call type 1, which has 1.5 nuclei per defect. And then we have type 2, which is 3.8 nuclei per cell division defect. This is a particularly interesting or fun defect that we like to call Mickey Mouse. And then also we have type 3, which is premature budding. So you have a cell here that is budding, and then you have this cell also budding before it is split off from the mother cell. So that is not found in wild type yeast cells. And then we have finally type 4, which is the most obvious cell division defect, when you have multiple budding sites and just a failure to split off from each other. So once I have these images, I'm just going to go through a typical way of how I would quantify this image. So I would count each cell division defect that I would see subjectively and record it on a chart in Excel. So in this image, I see one type 1, which is labeled here in the red, circled here in the red, and I will record that as type 1. And then I don't see any type 2, and it's just a side note, it's not very common to see every cell division defect in a single image. So I would also see one type 3 cell labeled here in the purple cell, and I record it here. I don't see any type 4, and then I see around 19 normals. So then I would take the percentage of those cell division defects and then give them their weight based on their nuclei per cell division defect. And then we have our actual data that we can use and start analyzing. So just to give you a picture of how extensive this project can be, there are 36 possible combinations of Epsin 2 and yeast tip 1 truncations. And in order to test for significance, these experiments need to be repeated 9 times. So that's 36 times 9, which is 324 experiments. And this image you see right here is just one image. Usually 9 or more images are required for just one data sample. So that's 9 times 324, which is 2,916 images for this experiment. So as you can see, that is a lot of images even for one person to go through. So over the summer, we were thinking about creating an automatic algorithm to determine cell division defect. And from here, Felix will take the floor.
All right, I'll be explaining the step-by-step process of underlying our algorithm. Here you see the actual underlying process within each step. Now, the experimenter will only see the image or images inputted and the outputted quantification. But I'll be going through each step in the upcoming slides. Oops. Do that. Okay. Why is it?
Don't swipe down. Stay in the center of the mouse.
That's right. Yeah. Okay. First, we take the ZVI image, which is an image format produced when McKeith or the researcher does a microscopy image, and we then convert that ZVI image to a PNG image. We chose to use a PNG image because it is a lossless image format, so we keep most of our data the same as the ZVI image. Now, in order to classify each cell, we must first extract each cell for classification. First what we do is called an O2 binarization. What this does is it produces a histogram that you see on your right. The upper level histogram pixels are the background, while the lower ones are the cells. Now, what O2 tries to do is tries to classify both and then separate one from the other. Now, once it does that, it produces a threshold that maximizes the variances of each section. In this example, we get a threshold of 131. So any pixel above 131 would be considered the background, while all the pixels below it would be considered the cell. And below you see the output. I'm not gonna go there. There's supposed to be an image right there. Next, we use breadth first search, which is a mapping algorithm. What this does is it goes through the image and then finds all the black pixels and then clusters them. And then what you see on your bottom right is the output of after breadth first search. And we do this throughout the cell and extract each cell that we have. Next, what you see on the top left is after a zoomed in image of after an O2 reduction, and on the top right you see the type 1 image. Within the O2 reduction, you see a little white space right here, and right here you see the extracell wall described by McKeith earlier, and over here you see that we lose this part of the information. Now, we began collaborating with Dr. Sharabadi in the statistics department, and he proposed using an algorithm he produced before for a different project. What it essentially does is it uses the Laplacian, which is the second derivative in the x and y direction, and then sums the pixels, as well as adding a weight that he had calculated earlier, and what we get is this picture below. Now, the best part of this image is that the boundary is now a solid boundary compared to the O2. There is still the problem within the cell. You see right here is that there are still boundaries within the cell. Now, to fix this issue, Michael, one of our collaborators, decided to create an algorithm called bounding. What he finds is the furthest x point on the boundary and the furthest x point on the right side of the boundary, as well as doing it for the y direction. And we produce a grid similar to the one you see right here on the right. And after this, any pixel that's contained in both the x and y direction is considered part of the cell. Anything that's considered in only one direction is considered part of the background. And afterwards, this becomes a solid image, which we can now find useful. Now, afterwards, once we have this, now that we have a solid image, we can take the average in the x direction and the average in the y direction. What this produces is called a centroid, which you can see right here. Afterwards, we find the furthest pixel away from the centroid that's still considered part of the cell. After this, we consider that the radius, and we draw a circle from that radius, as you can see from the following images. We next calculate a ratio from the area of the cell divided by the area of a circle. And as an example, we took an image from each type that we could find. So a normal, a type 1, a type 3, and a type 4. And here you see a histogram of our calculations. Some things to note are that on the top left, you see the normal cell ratios. It's a bimodal histogram, meaning that there are two peaks. The second peak, which is between 0.35 and 0.6, is produced by budding normal cell. On the top right, you see the type 1 cell ratios. It's pretty uniform, but most of the cells are contained above 4. On the bottom left, you see the type 3 cells, which are contained mostly below 3.5, as well as a few outsiders. Mostly those ratios are greater, usually because they're contained by nearby cells, and it's a problem we are currently addressing. On the type 4s, we see that most of the cell ratios are below 3.5. So what we propose is making 3.5, or an area between 3.5 and 4, as a threshold, so that we now group the cells. Normal cells and type 1 cells would be ratios above 3.5, and type 3 and type 4 cells would be ratios below 3.5. Next, we have to separate the normal cells from the type 1 cells once they are grouped away from the type 3 and type 4s. On the top left, you see a normal cell. Below that, you see a heat map of the cell. The first thing to note is that on the boundary, it is a pretty much uniform and solid boundary, while on the top right, you see a type 1 image and a heat map below it. Some things to note from this is that the boundary is not uniform, as well as a section right here, which is the extra cell wall described by Keith earlier. What we propose with Dr. Sharbati is using a combination of his detected edges that we used earlier to find the boundaries, as well as an O2 reduction or a different type of thresholding mechanism so that we can actually find the area of this extra cell wall. Since the normal is uniform, it should produce an area approximately 0, but a type 1 cell should produce an extra cell wall area much greater than 0. So any cell wall greater than 0 would end up being classified as normal, as well as any area greater than 0 would be classified as a type 1 cell. Now, on the left, you see a type 3 image, and on the right, you see a type 4 image. Now, using the centroid we found at the beginning, we proposed connecting the centroid to each boundary pixel on the type 3 and type 4s. From this, we will find the distribution of the distances between the centroid and the boundary points. One thing to note is that the type 3 cells are pretty uniformly dispersed right here, while the type 4s have really unusual shape morphologies. So what we propose is that the distribution of points will be less extreme on the type 3s and more extreme on the type 4s. Now, what we propose is having a threshold when the distribution isn't extreme, it'd be a type 3, and when it is very extreme, it'd be a type 4. We plan on getting a lot more images and then finding a threshold so that those above the threshold would be the type 4 and those below would be the type 3.
So to conclude, we are still currently working on developing an algorithm that we can confidently say will identify every cell division defect without there needing to be any user input. So we're trying to make this algorithm as automated and as user-friendly as possible. And so since ImageJ is the software that I currently use for image processing, we would think that it would be a cool idea for us to make a plugin for ImageJ so I can use it with the image processing system. So future goals. Our future goal is to continue to make a more accurate and reliable algorithm. As of right now, the algorithm accepts one stack, which is one image at a single particular resolution. However, if we take an image of, say, 3 stacks at 3 different resolutions, we can run the algorithm on each stack, and each stack will have their own thresholds as described by Felix. So this will allow it to be a little bit more accurate, and of course there'll be 3 times as many images, but since this is done through a computer, it's feasible now. So we can now have the most accurate and most confident cell division defect identifier as we possibly can have. And with that, I would like to conclude by thanking Dr. Claudio Aguilar for being Felix and he's our mentor, and he's definitely put in a lot of work and helped us with this presentation, and we thank him for his time. And also we'd like to thank the Center for Science of Information for allowing us to undergraduates to do basically graduate research. We appreciate you giving us this opportunity, and at that, we'd like to ask if there are any questions.
Yes, good, great talk, guys. Any questions?
Thank you.
See, I have actually a non-technical question, just for also posterity, because this is being recorded and it's likely others will watch this presentation down the road. From your own perspectives, Felix coming from the math end of things and McKeith from the biology, what advice would you give to other student researchers who might either not think about the possibilities of reaching out to a different discipline, or maybe they are thinking about that, but they're not quite sure how to go about it? If you guys have, from your experience of working together with this and other projects, that'd be really helpful.
Yeah, I guess I'd probably have to say get exposed to as much statistics and as much math as it pertains to your field of study as possible. There's no way you're going to ever outrun math or
Yes.
statistics, no matter how scary it is. So I would say, yeah, definitely get into it and work. And this project has definitely helped me understand, because I've only had one statistics class so far. It's definitely helped me understand those basic principles that my statistics class first taught me. So that's what I would have to say.
I'll say from a math and stat point of view, what we learn is pretty abstract. So this is actually a great opportunity in seeing how we can actually apply what we know. For example, the variances, we never actually see how it's applied. We just see the numerical computations. And here we get to apply this on thousands of pixels, and we get to reduce a problem that could take, like McKeith, a couple hours, and we get to see patterns that McKeith might not see, but we see as an interesting pattern. So
we also plan, this algorithm definitely will save me a lot of time in the future, because doing this sort of quantification just by subjective analysis, just me staring at it, has taken me quite a long time. And we were running this on a couple images, 30 images, and it did it within like a fraction of a second.
Yes, we calculated on, I believe, the first set of images he gave us, which was 72 images that contained approximately over 300 cells.
Amazing.
And it took less
Yeah,
than 5
so
minutes
this
to
is
run
kind
all
of the power of putting fields together, especially from the
Definitely.
data science perspective, and applying it like Felix was saying. So it's exactly the kind of thing we want to continue to promote here with our center. Anything else that either one of you would like to add? I don't see any other questions at this point. With that, I think we'll sign off, and thank you again very much, both McKeith and Felix.
Thank you.
Yeah.